Okay, so acetyl-CoA carboxylase, this forms the rate-limiting enzyme of the fatty acid biosynthesis or conversion of acetyl-CoA to the 3-carbon malonyl-CoA. It is a deciding factor whether the synthesis has to be taken place or not. But since once the acetyl-CoA is converted to malonyl-CoA, it's not having any other alternative. It has to go on with the synthesis. So they think twice for whether the synthesis has to be taken place or not. And that is again decided by this multi-enzyme complex acetyl-CoA carboxylase. And since it is a very much important enzyme, it is undergoing regulation through two mechanisms, either by means of allostericity control or by means of covalent modification. Again, this acetyl-CoA carboxylase, it is the mechanism with which it is undergoing control. And again, it is having two modes of regulation, a short-term regulation and a long-term regulation. That is, long-term regulation, the enzyme will be under the control for a long period, right? Already we call this long-term control, and it is under genetic control. That is genetic regulation. ഈ ഷോർട്ട് ടേം റെഗുലേഷനിലാണ് അല്ലോസ്ട്രിക് കൺട്രോൾ ആൻഡ് കോവാലന്റ് മോഡിഫിക്കേഷൻ ഒക്കെ നടക്കുന്നത്. ബിക്കോസ് ലോങ് ടേം കൺട്രോൾ എന്ന് പറഞ്ഞു കഴിഞ്ഞാൽ ഇറ്റ് ഇൻവോൾവ്സ് ചേഞ്ചസ് ഇൻ ദ amount of the enzymes brought about by the changes in the rate of synthesis and degradation. അതായത് it is under genetic mechanism. അത് genetic regulationലാണ് വരിക. അതേസമയം ഷോർട്ട് ടേം റെഗുലേഷൻ ഇറ്റ് ഈസ് ഹാപ്പനിങ് ത്രൂ ഔട്ട്. ദാറ്റ് ഈസ് വെദർ എപ്പോഴൊക്കെ കാർബോഹൈഡ്രേറ്റ്സ് കൂടണം, എപ്പോഴൊക്കെ പ്രോട്ടീൻസ് എക്സസ് ആയിട്ട് സർപ്ലസ് ആയിട്ട് എന്തൊക്കെ ബയോമോളിക്യൂൾസ് ഉണ്ടോ അതിനെയൊക്കെ എന്ത് ചെയ്യും, TG ആയിട്ട് അത്രയും ഇഫക്റ്റീവ് ആയിട്ട് സ്റ്റോർ ചെയ്യും. Clear. So all of them come under short-term control, and the short-term regulation is under the mechanism of allostericity modifiers and also by means of hormonal regulation. That is by means of covalent modification. And glucagon is a hormone which is responsible for always inhibiting the acetyl-CoA carboxylase. It always keeps the acetyl-CoA carboxylase in the inhibitory state, right? And again, it is carried out by phosphorylating. The inhibition of the acetyl-CoA carboxylase is carried out by phosphorylating the enzyme. This enzyme phosphorylate it is under inactivated state. That is inactive. How can the glucagon inactivate the acetyl-CoA carboxylase? It can be simply phosphorylated. Phosphorylate enzyme it is a kinase enzyme. So which is the kinase enzyme? It is the AMP protein kinase. That is AMP AMP adenylate cyclase. A mechanism we have already discussed, glucagon. So adenylate cyclase converts AMP adenylate to AMP. So this AMP protein kinase enzyme undergoes activation. Again, this AMP protein kinase is under the control of another enzyme which is known as AMP kinase, AMPKK. So this AMPKK is activated by actually the glucagon. But glucagon is supreme power. So glucagon carries out the activation of AMP kinase or AMPKK. So this AMPKK, when undergoes activation, it causes the activation of AMP kinase, that is AMPK. Once this AMPK is activated, what happens? It causes the immediate phosphorylation of acetyl-CoA carboxylase, converting it from the active form to the inactive form. So that is how the covalent modification is happening. And there is the allostericity modification. So allostericity modification naturally involves allostericity modifiers. And the allostericity modifiers, inhibitory factors, one fatty acyl-CoA, excess of fatty acyl-CoA ഉണ്ടായി കഴിഞ്ഞാൽ ആ ഫാറ്റി acyl-CoA തന്നെ പോയിട്ട് ഇതിനെ ഇൻഹിബിറ്റ് ചെയ്യാം. അതല്ലെങ്കിൽ എപ്പോഴും ഞാൻ പറഞ്ഞു NADH to NAD റേഷ്യോ വരുമ്പോഴാണ് ഫാറ്റി ആസിഡ് സിന്തസിസ് അവിടെ നടക്കണത്. അപ്പം ATP തന്നെ പോയിട്ട് വേണമെങ്കിൽ ഇതിനെ ബ്ലോക്ക് ചെയ്യാം, ഫീഡ്ബാക്ക് ഇൻഹിബിഷൻ, അല്ലെങ്കിൽ NADH പോയിട്ട് ബ്ലോക്ക് ചെയ്യാം. സോ ദീസ് ആർ ദ വേരിയസ് ടൈപ്സ് ഓഫ് മോഡിഫയേഴ്സ്, റൈറ്റ്? Yeah. Then fatty acid. Fatty acid itself can cause the inhibition. Citrate activator ആണ്, പക്ഷെ സിട്രേറ്റിന്റെ ലെവൽ അങ്ങ് കുറച്ച് കളയും. ഗ്ലൂക്കഗോൺ ആണെങ്കിൽ ഗ്ലൂക്കഗോൺ goes and reduces the level of citrate. കാരണം സിട്രേറ്റ് എക്സസ് ആയിട്ടുണ്ടെങ്കിൽ citrate can simply cause the IMM and enters into the cytoplasm and the pathway can be initiated. But what does glucagon do? Glucagon lowers the citrate level. So citrate activator ആണ്. It is not the inhibitor. Citrate is the activator, but citrate is kept under strict regulation. It is whenever there is excess of carbohydrates in the inhibition immediately the citrate level is lowered. Citrate lower naturally mitochondrial cross so citrate is an activator while fatty acyl-CoA is an inhibitor. right? So citrate, in order to cause the inhibition of acetyl-CoA carboxylase, what is doing? Citrate is the lowering of the citrate level is carried out. The quantity of citrate is lowered. Like PFK1 will be inhibited. So citrate becomes the precursor of cytosolic acetyl-CoA and signal for the activation of acetyl-CoA carboxylase. So citrate the value, quantity and concentration of then phosphorylation, that's we have already discussed. That is glucagon, epinephrine, etc. It carries out the phosphorylation. Phosphorylate ചെയ്ത് കഴിഞ്ഞാൽ ഈ enzyme is blocked. Insulin എന്ത് ചെയ്യും? Activate ചെയ്യുക ചെയ്യാം. So additionally, these pathways are regulated at the level of gene expression also. അതാണ് ഞാൻ പറഞ്ഞ long-term control. But long chain fatty acids is controlled in short term by means of allosteric and covalent modification, but in long term by change in the gene expression. That is at the genetic level. Then what enhances or what is the regulatory, what causes the activation of acetyl-CoA carboxylase? We have already discussed excess carbohydrates, then അതുപോലെ proteins, ഏതെങ്കിലും ബയോമോളിക്യൂൾസ് എക്സസ് ആയിട്ടുണ്ടെങ്കിൽ സർപ്ലസ് ആയിട്ടുള്ള എല്ലാ അമൗണ്ട്സും ഗ്ലൈക്കോജൻ ആയിട്ട് ഒരുപാട് സ്റ്റോർ ചെയ്യും, പക്ഷേ ഗ്ലൈക്കോജൻ സ്റ്റോർ ചെയ്യുന്നതിന് ലിമിറ്റ് ഉണ്ട്. അപ്പം എക്സസ് ആയിട്ടുള്ളൊക്കെ ഇവിടെ TG പോയിട്ടാണ് സ്റ്റോർ ചെയ്യുക. I mean TG ആയിട്ട് adipose ടിഷ്യൂവിൽ പോയിട്ട് സ്റ്റോർ ചെയ്യും. So excess carbohydrates is stored in the form of fat in the anticipation of periods of caloric deficiency. എപ്പോഴെങ്കിലും സ്റ്റാർവേഷനോ ഹൈബർനേഷനോ അല്ലെങ്കിൽ മാൽന്യൂട്രീഷനോ എന്തെങ്കിലും വരികയാണെങ്കിൽ അപ്പം എന്ത് ചെയ്യും? TG comes into action. TG undergoes lipolysis. അപ്പൊ എപ്പോഴെങ്കിലും ആവശ്യം വന്നെങ്കിലോ വെച്ചിട്ട് നമ്മൾ എപ്പോഴും സ്റ്റോർ ചെയ്ത് വെക്കുക. And in between meals. Okay, nutritional state of the organism is a main factor in regulating the lipogenesis. So fed state, where that is when a person is in the well-fed state anengil, and if a diet contains a high proportion of carbohydrates, then the excess of carbohydrates will be entering into fatty acid biosynthesis. That inhibitory factor is lifted up. Inhibition lifted that it is undergoing activation. The acetyl-CoA carboxylase will be converted from the phosphorylated form to the dephosphorylated form. Lipogenesis converts surplus glucose and intermediates such as pyruvate, lactate, acetyl-CoA, etc. into pyruvate, lactate, etc. into fat by getting converted into acetyl-CoA. And when again lipogenesis എപ്പോഴാ കൂടുക? It is increased when the sucrose value is elevated. അതായത് വല്ലാതെ ഷുഗർ കഴിക്കണം. അതുകൊണ്ടാണ് വല്ലാതെ ഷുഗർ കഴിക്കുന്നവർക്ക് ഒരുപാട് തടിച്ചിരിക്കാനുള്ള കാരണം glucose, because bypasses the phosphofructokinase control point in glycolysis and floods the lipogenetic pathway. PFK1 നെ അങ്ങ് bypass ചെയ്യും. Now what happens during fasting? Fasting in the സമയത്ത് എന്താണ്ടാവുക? The acetyl-CoA carboxylase will be inhibited and depressed by restricted caloric intake, deficiency of insulin ഉണ്ടാവും. അപ്പം അങ്ങനെയൊക്കെ വരുന്ന സമയത്ത് increased concentration of plasma-free fatty acids, and an inverse relationship has been demonstrated between hepatic lipogenesis and concentration of serum-free fatty acids. That's not that important. What is the role of insulin? Insulin stimulates the lipogenesis by increasing the acetyl-CoA carboxylase activity, and it enhances the transport of glucose into the cell. It increases the availability of both pyruvate for fatty acid synthesis and also glycerol 3 phosphate. കാരണം fatty acid synthesis നടന്നു കഴിഞ്ഞാൽ it is always followed by TG formation. അപ്പോൾ TG ക്ക് എന്ത് വേണം? glycerol 3 phosphate വേണം. So what does insulin do? Insulin carries out the activation of both the acetyl-CoA അല്ലെങ്കിൽ fatty acid synthesis activate ചെയ്യും. അതേ സമയം തന്നെ it also carries out increased concentration of glycerol 3 phosphate so that TG can be synthesized. Then what happens? Insulin converts the inactive form of PDH to the active form, but not in liver, and thus provides acetyl-CoA. Insulin also acts by inhibiting cyclic AMP mediated lipolysis. Lipolysis അവിടെ ബ്ലോക്ക് ചെയ്യും. ഇതൊക്കെ പറഞ്ഞത് തന്നെ പിന്നെ തിരിച്ച് പറയുന്നതാണ്. Okay. Now next is fatty acid chain elongation. So this is how the regulation is happening. So the regulatory enzyme is the acetyl-CoA carboxylase. It's not the fatty acid synthase. Now, what is happening in the cytoplasm? The acetyl-CoA molecules, except one, all the remaining one will be converted into malonyl-CoA, and all these malonyl-CoA molecules condenses with the acetyl-CoA and the lengthening fatty acids. The elongated fatty acids undergoes condensation, and finally it gives rise to C16 palmitic acid, right? C16 carbon fatty acid will be synthesized. So Once this C16 palmitic acid is formed, then the cytosolic process will be stopped, right? If you are in need of a very long chain fatty acid, we know that the very long chain fatty acid cannot be synthesized in the cytoplasm. It's always processed in the microsomes. Fatty acid oxidation it is carried out by the very long chain fatty acids are processed first by the microsomes. And there or the peroxisomes where they are undergoing oxidation to give rise to long chain fatty acids. Similarly, in case of the synthesis also, once this long chain fatty acid is formed, C16 palmitic acid formed, if it is in need of, if the body is in need of the synthesis of very long chain fatty acids, then it is taken into the microsomes, right? That is rough endoplasmic reticulum. It is carried out, it is carrying out the chain elongation. So very long chain fatty acid synthesis are synthesized in the smooth endoplasmic reticulum, that is microsomes. It is happening in the microsomes, right? So next we are when we were discussing the introduction on the fatty acid biosynthesis, we have discussed that there are three modes of fatty acid biosynthesis. One is a extra mitochondrial system, അതാണ് നമ്മൾക്ക് പറഞ്ഞ cytosolic fatty acid biosynthesis. രണ്ടാമത്തൊന്നാണ് microsomal chain elongation, and the third one is mitochondrial chain elongation. So these are the three different mechanisms with which the fatty acid biosynthesis is happening. There is an extra mitochondrial system, that is a usual biosynthetic pathway. Then there is another one which is known as a microsomal chain elongation and mitochondrial chain elongation. So what do you mean by microsomal chain elongation? Microsomal chain elongation is nothing but the extension of the palmitic acid to give rise to very long chain fatty acids, which is happening in the microsomes and carried out by microsomal chain elongase system. Microsomal chain elongase system. So the reaction involves all the same step which was happening in the cytosol also, that is condensation, reduction, dehydration and reduction. But it is not the fatty acid synthase that is carrying out the elongation. Fatty acid synthase is present only in the cytoplasm. In the microsomes, right, in the smooth endoplasmic reticulum or in the mitochondria also, the fatty acid synthase complex is not there, or the fatty acid synthase, the elongation and the is happening, carried out, even though the reactions are the same, the enzyme is not present in the form of a multi-enzyme complex. They are individual enzyme. Condensation reaction നടത്തുന്നുണ്ട്, reduction carry out ചെയ്യുന്നുണ്ട്, again dehydration is carried out, and also the second round of reduction is also taking place. Even though the reactions are the same, the enzymes are not acting in the form of fatty acid synthase. It's not a multi-enzyme complex. They are individual enzymes. So there are three mechanisms with which fatty acid biosynthesis are taking place. There is an extra mitochondrial system carried out by fatty acid synthase complex. Then there is a microsomal chain elongation which is carried out by microsomal chain elongase enzyme. So there also the substrate is a malonyl CoA. So the acetyl CoA is coming in the form of malonyl CoA, and malonyl CoA undergoes condensation with the C16 palmitic acid. It first condenses with the removal of carbon dioxide. Then we are having a beta keto acyl enzyme. Then the beta keto acyl group is converted into the dehydrated form, right? Then it is converted into the next reduction. So all the reactions are happening in the same way as that of in the case of beta oxidation. The NADPH is the reductive reduction factor also. The protons are provided by the NADPH only, but the enzymes are acting in an individual manner, right? So they are separate and they are not clustered like that of a fatty acid synthase complex, right? So the malonyl CoA is a substrate molecule. It simply gets added on to the palmitic acid, right? The palmitic acid moves or migrates from the cytoplasm into the microsomes where it is undergoing elongation, and the elongation is carried out by malonyl CoA in presence of NADPH, right? And the chain is getting elongated, right? And molecular and oxygen is also necessary because NADPH is the reaction, is a coenzyme. So it requires molecular oxygen also, right? Then there is another type of elongation which is happening is in the mitochondria. Because mitochondria is, even though it is a part of a cell, it's having its own machinery. Mitochondria ke vendittulla lipids okke synthesize cheyna mitochondria thaneyana. Clear? So it is a mitochondrial chain elongation is carried out by another enzyme system known as mitochondrial chain elongase. Pakshe avade it is not the malonyl CoA. The reaction is carried out by acetyl CoA itself, not the malonyl CoA. The acetyl CoA adupole NADPH will be replaced with NADH. Clear. And also ATP. So these are the differences. It's entirely different from that of the cytoplasm or the extra mitochondrial system and the microsomal system. It is making use of acetyl CoA, NADH and ATP.